Hello everyone, welcome to my YouTube channel Zoologic Minds. On this channel, I'll be covering topics related to zoology and life sciences. This is my first video on this channel, so without any waste of time, let's get started. Our topic is classification of animal kingdom. Why do we need to classify animals? For the ease of study, to know the diversity and identifying the phylogenetic relationships, etc. The kingdom Animalia is divided into Mesozoa, Parazoa, and Eumetazoa. The Parazoas include phylum Porifera, and Eumetazoas are further divided into Radiata, Bilateria. The Radiatas include phylum Cnidaria and phylum Ctenophora. The Bilaterias are further divided into acoelomates, pseudocoelomates, and coelomates. Acoelomates are flatworms, that is phylum Platyhelminthes. The pseudocoelomates are phylum Aschelminthes, and coelomates are Annelida, Mollusca, Arthropoda, Echinodermata, Hemichordata, and phylum Chordata. The phylum Chordata is further divided into Urochordata, Cephalochordata, and Vertebrata. The Urochordates include notochord in their tail, and Cephalochordatas have notochord from tail to head. And subphylum Vertebrata is also known as Craniata. They are divided into Ignatha and Gnathostomata. Ignatha do not have jaws in their mouth, and Gnathostomatas have jaws in their mouth. The Gnathostomatas are further divided into superclass Pisces and Tetrapoda. The Pisces have class Placodermi. They are extinct fishes. The class Chondrichthyes, cartilaginous fishes, and class Osteichthyes, they are bony fishes. The superclass Tetrapoda includes class Amphibia, class Reptilia, class Aves, and class Mammalia. So in upcoming classes, we will discuss about each phylum and class in detail. First, let's know what are the basis of classification. The first basis of classification is level of organization, that is cellular level, tissue level, organ level, and organ system level. In the cellular level of organization, the body consists of many cells which are may or may not be similar in function. For example, sponges, they have cellular level of organization, and then tissue level. This is the aggregation of cells. And this type of organization is in cnidarians and ctenophores. The next is organ level of organization, and platyhelminthes have organ level of organization. And then organ system, from Aschelminthes to chordates, they all have organ system level of organization. The second is body symmetry. The body symmetry is if we are dividing an animal from a plane, if the body is divided into two equal halves, they are known as symmetric animals, and if their body is not divided into equal half, they are called as asymmetric animals. Thus, the symmetry can be radial symmetry or bilateral symmetry. In radial symmetry, we can divide an animal into two equal halves from many planes. For example, in sponges, cnidarians, and echinoderms, radial symmetry is found. And in bilateral symmetry, the animal is divided into two equal halves, but from a single plane only. And this type of symmetry is found in all vertebrates and many invertebrates also. The germinal layers, it can be either diploblastic or triploblastic. The diploblastic animals have only two germinal layers, that is ectoderm and endoderm. The ectoderm is outer layer and the endoderm is inner layer. Poriferans and cnidarians are diploblastic animals. The triploblastic animals have three germinal layers: outer ectoderm, inner endoderm, and mesoderm is found in between these two, ectoderm and endoderm. And the triploblastic animals are from platyhelminthes to chordate. Body segmentation. It is the differentiation of body into distinct portions. So it can be either metameric, that is true segmentation. In this type of segmentation, the external divisions are corresponding to the internal divisions. For your body organs are arranged according to the external segmentation. And this type of segmentation is found in annelidas, arthropodas, and chordates. And pseudo metamerism means false segmentation. That means the external segmentation is not according to the internal segmentation. This type of segmentation is in Teniasolium flatworm. The body is divided into different proglottids: immature, mature, and gravid. Body coelom. Body coelom is the fluid-filled cavity of an organism that contains internal organs. They can be either coelomates, pseudocoelomates, and eucoelomates. In acoelomates, they do not have any coelom. These are sponges, cnidarians, and ctenophores, and flatworms, platyhelminthes. Pseudocoelomates are, they have a body cavity, but it does not arises from mesoderm. That is why they are called as pseudocoelomates. They are the roundworms are pseudocoelomates, and eucoelomates, they have true body cavity and it arises from mesoderm. That is why they are called as eucoelomates. And it can be further divided into schizocoelomate and enterocoelom. Now, the schizocoelom develops from the splitting in the mesodermal layer. For example, in Annelida, Arthropoda, and Mollusca, the schizocoelom is seen. In enterocoelom, the body coelom is formed from the out-pocketing of the mesodermal layer, and this type of coelom is found in hemichordates, chordates, and also in echinoderms. Thank you for watching my video. Stay tuned for the next video.